So let me first of all thank the organizing committee for having me here. It's my first time. I'm delighted to be here. I'm more a lymphoma expert, but I hope that the things that more or less I'm going to convey to you today will be as general as possible so that we actually can all together contribute to understand better how the receptor contributes to malignant B-cell transformation and maintenance. So since I'm the first in this session, just a very, very brief summary of what the receptor is actually doing in normal B cells. So as you all know, B cells start from a progenitor that undergoes a number of somatic rearrangements that actually have the ultimate goal to express on the surface a functional immunoglobulin receptor. Now this receptor, of course, signals through B cell development, and the signals that it delivers to B cells changes according to the stage of differentiation. So if the strength of the signal, for example, is too strong in a developing immature B cell, that cell will actually either undergo apoptosis or will eventually become anergic. If the cell are allowed to exit this first checkpoint, they are actually allowed to continue further in their differentiation, giving rise to a number of different B cells with different actually functions and where the signal strengths delivered through the receptor is actually different. So the fate that the cell acquires as a result of signaling coming through the receptor is something that is very important to define the identity of these cells. And there are a number of different experiments in literature, mainly in mouse models, where this has been, in the meantime, confirmed. Now, another important moment in the life of a lymphocyte is when it recognizes antigen and gets recruited in the germinal center. This is an amazing Darwinian environment where very few cells survive. These are the ones that acquire somatic mutations, and as a result of those mutations, they increase the affinity for the antigen. So the receptor is, again, a key element here in deciding life and death of a B cell, and the signal through this receptor will also instruct whether the cells will eventually differentiate into a plasma cell or into a long-lived memory B cells. So this is basically just to introduce you how important this receptor is for any type of decision that the B cell encounters through its life. Now, since we're in a CLL meeting and since we're in a tumor-related environment, I'm just trying to now translate this information to a tumoral setting, where actually we are depicting here different types of tumors that are the result of oncogenic events that are actually occurring at specific stages of B cell development. A vast majority of tumors have experienced the expression of AID. We heard about AID before. AID is, of course, important for humoral immunity, but it's also a major cause of mutations and chromosomal translocations that actually drive a number of different B cell malignancies. But nevertheless, despite all these different genetics that are underlying the different type of tumors, the majority of mature B cell tumors, except Hodgkin disease, are actually tumors that still retain a B cell receptor on the surface. And the question has been for many, many years of what is the receptor actually delivering to a malignant cell that has already p53 mutation, has a strong MYC overexpression, or has a BCL2 overexpression? How could the receptor still contribute to controlling the survival and the growth of these cells? And I just put here, and I apologize, this is just an historical perspective. Every one of you is probably familiar with that. There are a number of different genetic evidence that hinted to the role of the receptor being critical for malignant transformation of B cells. One is actually just looking at the pattern of somatic mutation in the variable region genes in those tumors, of course, that experience the germinal center, with basically very few mutations accumulating in the framework regions of this immunoglobulin, suggesting the importance of retaining a functional integrity of the receptor in the tumor cells. You all know that chromosomal translocations featured in a number of different tumors usually attack the non-functional chromosome of the immunoglobulin heavy or light chain loci, pointing to the fact that probably the receptor is important in the initial phase of malignant transformation. And of course, in this environment, I don't need to point out that actually the identification of unmutated and mutated subtypes of CLL had already hinted, and the identification of a number of stereotypic BCRs, that actually the B cell receptor was indeed recognizing potentially something and actually driving malignant transformation or maintenance. So this is just to summarize what we know so far about how B-cell receptors talk to the environment. So there are, of course, a number of evidence, and many of them have been contributed by people in this audience, indicating that foreign antigens, bacterial pathogens, but also self-antigens or homotypic interactions are all potential mechanisms through which the B-cell receptor can activate a signal in tumor cells. And the genetics that has followed, actually, biochemical studies has also indicated that specific mutations affecting specific components of the B-cell receptor signaling pathways are also in some tumors constitutively activated, pointing further to the importance of this pathway for malignant B cells. Now, the real killing experiments that actually provided a big push to the identification also of drugs interfering with B-cell receptor signaling and to bring them in the clinics were a number of different experiments done in the beginning of 2000 by the Stout group and others, where they actually knocked down proximal components of the B-cell receptor in different types of tumors, and showing actually that this was very effective in killing a majority of different tumor cell lines. This has led to a race in the identification of a number of inhibitors that are in the meantime being actually introduced in the clinics, targeting a number of different effectors of the proximal part of the B-cell signaling pathways, which are actually giving amazing results in the treatment of CLL, mantle cell lymphoma, and a few other diseases, but there are more and more to come. Now, despite all this, and despite this evidence and the fact that these inhibitors indeed are able to actually treat patients, we still know quite little about the biology of the receptor in these tumors. What does this receptor finally really delivers to a tumor that has already so many other mutations? And this was basically the purpose of what we have been doing in the past 10, 12 years, trying to understand how the B-cell receptor delivers survival and proliferative signals. And we started addressing this in mouse models. So we basically engineered, and I apologize, this is not an aggressive lymphoma audience, but basically I believe that many of the findings that we made here are relevant for this auditorium. So what we did was to engineer the mouse model where we actually combined the MYC oncogene, which is the hallmark of a very aggressive B-cell lymphoma, Burkitt lymphoma, with a conditional heavy chain allele, so that we could inactivate by Cre-Log-Sp the expression of the B-cell receptor at any time point during tumor transformation. And that's a very efficient process. You can actually isolate tumors from this animal, or you can inactivate the B-cell receptor directly in the lymph node or in the spleen. And what you do is you basically very easily identify B-cell receptor negative cells by flow cytometry as they actually lose surface IgM expression. Now, the surprising result was that actually when we did this experiment, we repeated this experiment multiple times, the result was always the same. Tumors without the receptor could easily grow in vitro, and when we would transplant them into immunoproficient animals, so with animals that have their own immune system, these tumors were able to kill the animal very rapidly. There was still a difference between BCR positive and negative tumors, but the message is that actually tumors, at least this type of tumors, can actually evade the B-cell receptor in order to continue growing. And this was a bit baffling because of all the experiments had been done before in the literature on knockdowns in cell lines. So we started looking at these papers a bit better, and we realized that in every single paper, what the authors did was actually to knock down the B-cell receptor effectors, but keep the mutant cells in competition with B-cell receptor expressing cell. So when you do the same trick here in MYC transformed cell, you see a very different picture. So you see that upon B-cell receptor deletion, following the cells in vitro over time, the only cells that actually remain in the cultures are the ones that retain the B-cell receptor, indicating that the receptor is per se not important to keep the cell surviving, but provides a critical fitness advantage to the tumor cells. And this is also seen when you transplant mixtures of 50-50 in vivo, and basically the only cells that you can collect back from very affected mice are actually cells that retain the B-cell receptor. So the receptor is important for tumor cells, but it's actually important to provide an advantage in the context of a progressing disease where basically you could lose, in principle, B-cell receptor expression during evolution, for example, as a result of somatic mutation. And in order to immediately validate these findings in a human settings, we collaborated with a number of pathologists in Milano and Brescia, Maurilio Ponzoni and Fabio Facchetti, and started staining sections of Burkitt lymphoma for the B-cell receptor, in this case IgM, but we actually stain for all different isotypes. And this is a representative case where in the same tumor parenchyma, we have cells that retain the B-cell receptor and other cells that actually, by the way, have already lost the B-cell receptor. As any tumor, if you keep this tumor evolving enough, they will eventually acquire secondary mutation that will restore eventually fitness to the cells. And this is a typical example. So if we grow tumor cells that have not seen B-cell receptor positive cells in their life for a certain number of weeks, both in vitro and in vivo, they will select and pick up some secondary mutation without any pressure. But these mutations, some of them will be enough to actually restore full fitness of the cells. And this is telling you that tumors on the way can acquire secondary mutation, becoming totally independent of the B-cell receptor, even in competitive settings. So we worked out the signaling pathway. I have no time to actually show it. It has been, in the meantime, published. But basically, what we could show is that this fitness signal is very much dependent on a PI3 kinase AKT JSK3 beta axis, whereby when you lose the B-cell receptor, you weaken this axis, you activate stronger JSK3 beta, and this activation has a negative impact on MYC-controlled gene expression. So there's a very linear relationship between expression of the B-cell receptor, in this case tonic B-cell receptor signaling, because this is a model that does not entail any recognition of foreign antigen, and actually a direct regulation of the oncogene that drives the disease. If you wait long enough, tumors will select secondary mutations, and whole exome sequencing experiments in this mouse model revealed that the MAP kinase pathway is very commonly affected as a way that allows tumor cells that have lost the B-cell receptor to fully restore their actually fitness capacity, feeding back into the activation of the MYC transcriptional program. Now, how is actually the receptor providing this competitive fitness? So one possibility, which we already validated, was basically controlling the metabolic state of the cells. So what we did here was to label cells with actually either glucose, pyruvate, or glutamine, and follow catabolism of the sugar and the amino acid, trying to find out how the loss of the B-cell receptor affected actually central carbon metabolism. And I show you two independent experiments. So tumors can behave in different ways. In one case, we identify tumors where the loss of the receptor leads to an adaptive higher consumption of exogenous glucose. In another case, actually, the loss of the receptor has actually a negative impact to the glutaminolysis, which is a major driver of central carbon metabolism in MYC-transformed cells. So depending also on the genetic background, the role of the receptor on the regulation of central carbon metabolism can be different. Now, is this only a mouse model? I showed you already before that we did some preliminary staining in some sections of Burkitt lymphoma. We're actually aggressively following up on this, staining actually all sorts of aggressive B-cell tumors. And I'm showing you just an example of a Burkitt lymphoma where the entire tumor has lost B-cell receptor. And the reason for that is because they acquired a crippling mutation in the clonal VDJ rearrangement that prevents the expression of the heavy chain. So there is indeed evidence in human cancers that this process can happen. And if we look more also now in diffuse large B-cell lymphoma, which is another germinal center tumor, we have over 20% of these cases where there's no detectable BCR expression. So, you know, in order now to rationalize what we saw so far, in order also to discuss a bit what this could mean in terms of also treatment with B-cell receptor signaling inhibitors, what we basically envision is a scenario that actually the clinics will tell us whether it's actually true or not. Namely, in a progressing tumor, there will be always some cell losing stochastically B-cell receptor expression. These cells will be kept under control by the cells expressing the receptor. However, if you start hitting the B-cell receptor positive cells with anti-BCR inhibitors, you might unleash the negative clones, which will actually give rise to secondary tumors. You might, of course, also have situations where the activation of downstream pathways like the RAS-MAP kinase pathway could render the tumors completely independent of the B-cell receptor. So these are the questions that we are actually now working on quite intensively. So the first question is really, how are tumors able to overcome the acute loss of B-cell receptor? I mean, what is actually the biochemical signal, and what are the survival signal that sustain the growth of these cells? we actually, is the importance of the receptor in different tumors different? In other words, are the signals that the receptor is providing different, so that the mechanisms that allow a cell to evade B-cell receptor expression, could they be also different in different tumors? And could this help us to actually find better treatment for cases showing receptor loss or resistance to B-cell receptor inhibitors? So I'll give you a few hints of what we have seen so far. So one hypothesis was that in B-cell receptors negative cells, the cells still survive because they still have a CD19 receptor on the surface, which feeds into the PI3 kinase delta pathway, which is important for a number of tumors, including CLL. So we did a very simple experiment. We took these tumor lines, we knocked out first CD19, and then actually also the B-cell receptor, and asked the simple question whether the combined loss of BCI and CD19 was actually finally killing the tumor cell. And the answer is no. I mean, the tumors can easily evade both B-cell receptor and CD19 expression, and we'll see in a second why. And the reason is basically because they become resistant to PI3 kinase delta inhibition. So they are probably rewiring their signaling pathway. So you can see here that BCR knockout cells are quite resistant to idelalisib. And if you see actually the knockout cells are actually more resistant than the CD19 positive B-cell receptor positive cells, and all of them actually are quite resistant. So they are very likely using different pathways. This is basically what we are trying to hypothesize now. So it's either a different isoform of PI3 kinase that comes into play, possibly downstream of a different receptor. It could be a growth factor receptor. Of course, it could be also an epigenetic rewiring, and we heard a lot this morning coming from microenvironment signals or genetic mutations, and all this is being investigated. But one thing that we did already was to actually start tracing the evolution of a tumor cell that actually undergoes B-cell receptor loss. So what we did was to isolate tumors at different stages of the loss of the acquired loss of the B-cell receptor and profile them at the transcriptome level, looking at single cells, trying to really find out whether there is an evolution of these tumors. And this is just one representative example where we profiled something like 600 tumor cells. When we actually associated the genotype to the transcriptome and to the clustering, we recognized that there were distinct clusters. One cluster contained all the BCR positive. One other cluster contained the BCR negative that acutely lost the B-cell receptor, whereas these cells are the ones that acquired RAS mutations and were further down in the evolution trajectory. And we could just through transcriptomic analysis identify two major branch points. So one point where B-cell receptor positive cells become negative and become fully independent of the receptor, and then a second one where actually acquire RAS mutations and they finally become also fit again. And there are, of course, a number of signatures that we have identified in these two moments of B-cell receptor loss evolution, and we're trying to find out what are the main mechanisms that actually control this evolution. One thing that I wanted also to touch upon, because it was actually shown a lot in the morning. So there's been wonderful talks in the morning on the role on how the epigenetics of CLL is actually influenced by the oncogenic events that drive the disease. The B-cell receptor is one of them, of course. So it would be interesting to understand how much of the epigenetic profile that the CLL has is the result of signals through the BCR rather than through other signals coming from either the microenvironment or from the genetic setting of the tumor. What we did here was to really, in this model, knock out the B-cell receptor and profile the epigenetic landscape of these cells to understand how much of the signal coming through a tonic BCR is affecting genome-wide, the opening of loci, the opening of enhancers. And this is a very preliminary study. Here we are just mapping one histone mark that is usually associated with transcriptional activation, both at promoters and enhancers. And basically what we discovered is that actually when you lose the B-cell receptor, there are a number of peaks that are being acquired specifically in BCR negative cells, especially at regions that are actually called so far by a number of different algorithms and programs and different approaches like the Phantom5, which measures eRNA and RNA expressed from enhancers. We could see that actually the loss of the B-cell receptor actually has a unique feature, a unique set of enhancers, that putative enhancers that are being actually modulated by the lack of the receptor. And you can appreciate here we are representing just a number of different intergenic regions, so we are not focusing here on promoters. And we can see that in some regions, the receptor loss leads to actually a marked reduction of H3K27 acetylation. In some other regions, you have actually an increase. So it is very likely that actually the receptor has an important impact on the regulation of gene transcription, not only at the promoter level, but also at enhancer level. And this is something we are actually very interested to pursue. The single cell analysis identified among the pathways that were enriched among the cells that were losing the B-cell receptor, a reduction in a number of different ribosomal proteins. So we tested whether actually the fitness of the cells was indeed related to any problem in a protein anabolism. So what we did here was a very simple experiment. What you can take tumor cells, and they can be either positive or negative for the receptor, and actually treat them transiently with puromycin. So puromycin mimics an amino acyl tRNA. It's incorporated in nascent proteins. And then you can actually fix, permeabilize, and stain with antipuromycin and measure the amount of proteins that are being synthesized in the interval between actually the staining and the fixation of the cells. And we did this in many different tumors, so you can then sort on BCR positive and negative cells. And in every single experiment that we did so far, we observed a reduction in the amount of protein synthesized by tumor cells once you lose the B-cell receptor. And this explains also another important finding that we believe could be important for the clinics, namely that actually if you start hitting tumors or mixtures of BCR receptor positive and negative cells with an mTORC inhibitor, so an inhibitor of a pathway that is controlling actually protein synthesis as a result of amino acid availability, we could actually show that if you lose the B-cell receptor, these cells are much more sensitive to mTOR inhibition. Another important thing that we basically had to prove is that those tumors that restore fitness through, for example, MAP kinase activation, they could actually get killed by inhibiting this pathway. And this is what we have shown here. So we are showing here a clone that has acquired a RAS gain of function mutation in the NRAS gene. This mutation does not allow, so allows the cells to compete well and actually outcompete the B-cell receptor positive cells. If you start treating these tumors with inhibitor of the MAP kinase pathway, we can actually have a very dramatic effect. So the tumors are becoming dependent on this, addicted on this pathway. Another thing that actually we also observed quite consistently is basically a regulation that the receptor imposes on DNA damage responses. So again, I appreciate very much the talk of the former speaker. We don't know exactly the pathway that controls this, but every time we lose the B-cell receptor, we increase the activation of the DNA damage response, looking at, in this case, at phosphorylated H2A, but we looked at that measuring 53BP1 foci. And the interesting thing is that this process is totally reversible. So if B-cell receptor negative cells are complemented back with another receptor, you again optimize the DNA damage response, or at least the cells can cope better with the underlying. So these are tumors that have not seen any drug. They probably are suffering from replication stress imposed by MYC. And this replication stress is very much influenced by the expression or the lack of expression of the B-cell receptor. And this creates also an opportunity, because if you now combine positive and negative cells and start irradiating the cells with sublethal doses of ionizing radiation, you can see that you can appreciate that the B-cell receptor negative cells are more sensitive to ionizing radiation than the positive cells. Finally, we spoke also this morning, I heard already, and I think there will be more talks talking about the relationship between tumors and the microenvironment and how the immune system recognizes potentially tumor cells. We believe that the B-cell receptor is an important regulator of immune tolerance. This is just an example to show that when we delete the B-cell receptor, we actually have a pretty marked downregulation of PD ligand on the surface. We have also an upregulation of a number of NK ligands for activating receptors. So one possibility that would explain why in most of the tumors you still have a persistent major population of BCR positive cells is because while they are generated during tumor progression, they might be constantly eliminated by the immune system unless they select secondary mutation. So with this, I leave you with our view of what needs to be done to better understand how the B-cell receptor actually controls life and death of a malignant B cells. And I hope this can be applied also to CLL. We are actually working on that as well. So of course, on one hand, we would like to understand the mutations and epigenetic changes that are associated with acute and chronic loss of the B-cell receptor, especially those cells that actually can compete well with B-cell receptor positive cells. We want to identify biomarkers, because so far what we're doing is staining sections with immunoglobulin, with antibodies against immunoglobulins. We would like to identify a number of markers that could predict a diagnosis if there is a fraction of B-cell receptor negative cells, and these would be very dangerous cells, especially if the patient receives anti-BCR drugs, because they would respond less to the therapy. We are interested in understanding which drugs can specifically kill BCR negative cells, and maybe this could be used in a combined fashion with anti-BCR inhibitors, and importantly, work with our in vivo model, which allows us to study in immune competent setting to understand the interaction between adaptive and innate, contact with adaptive and innate immune cells, the migration of cells through lymphoid organs as a result of B-cell receptor expression, and the communication with trauma cells. So with this, I leave you with my most important slide. So this work is really the result of 12 years of intense work by many people in the lab, some still in the lab, some others have already in the meantime left. A number of critical collaborators, Maurilio Ponzoni at the San Raffaele Hospital, and Fabio Facchetti at Brescia. Without those pathologists, this project would have never been able to move from the mouse model to the human setting, and a number of other important collaborators. I would like to thank in particular Klaus Rajewski, with whom I did my postdoc and where I started this work almost 15 years ago when I was still at Harvard, and the funding agency. And with this, I'll be happy to take any question.
Thank you very much, Dr. Casola. We have about 4 minutes for questions. Are there any questions from the audience, please?
Francesco.
Thank you very much. Just one simple question from my side. How much of the B-cell phenotype is maintained in your model, and how much does it remain of the Burkitt lymphoma-like phenotype in the models, in the primary samples determined by Maurilio?
So you mean in terms of phenotypes?
Phenotype, either gene expression or protein
so,
profile.
I mean, for the mouse model, we have already shown in our paper 2 years ago that actually the major signature that changes when you lose the B-cell receptor is a very unique MYC control signature. It's not every single gene that is controlled by MYC that is deregulated. So we are trying to understand why only a subset of MYC controlled genes is being affected. We don't have major changes in the identity of a B cell. The B cell remains with its identity. It has the same type of transcriptional networks that sustain the life of a mature B cell. Concerning the human setting, we have yet no fresh material on which we can understand how the transcriptome or the epigenome of this cell has changed. So this is another thing we're actually aggressively doing. One thing that I would like to stress, because I think it's important also in the context of which is the normal cellular counterpart of a specific tumor, I very much agree with what has been said this morning. So I think the signals that are delivered through the receptor change very dramatically, whether you're a marginal zone, a follicular, a germinal center, light zone. So I think trying to find the correlate of the disease means that we have to really go much deeper than just taking naive B cells. We will have to take very specific subsets and try to be more and more detailed in defining those. I mean, I think the experiments this morning have already confirmed that actually most of the epigenome of a CLL comes from the original cell.
We have one more question, please.
Thank you. Thanks a lot for that excellent overview. So considering also what was shown this morning, that CLL cells at least also show genes that are specifically expressed more by T cells. I was wondering whether when you are deleting now your B cell receptor, whether you also find genes that are more, let's say, expressed rather by T cells, maybe also genes that are relevant for T cell metabolism?
Yeah, so actually we have some signaling pathways that are shared between B and T cells, but it's hard then to say whether these are T cell associated or it's still something that remains between the two. So my short answer is that we still haven't looked carefully enough to really reach any conclusion.
Okay, if there are no further questions, thank you very much, Dr. Casola. We're going to move on.